Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, in the previous class, we have started with the human genetic disorders, and in which we have covered the topic that is chromosomal disorders, which are not going to inherit in the offspring from the parent, means which are the non inheritable or not inheritable disorders. But today we are going to talk about the Mendelian disorders. And these Mendelian disorders are the gene related human disorders. Okay, so we are going to discuss this. So, if you talk about this Mendelian disorder are gene related human disorder. And if we talk about this gene related human disorder, you will see that this occur due to mutation in sickle gene, due to mutation in single gene, and they are transmitted to the offspring as per Mendelian principle. So they follow Mendelian Mendelian inheritance pattern. So these are inheritable disorders, and they can be traced by pedigree analysis. Okay, so this is the particular thing. Now the common Mendelian or gene-related human disorders are of different type. They can be due to mutation in the autosome, or they can be due to mutation in a, we can say, a allosome or sex chromosome. But if mutation occur in autosome, they can be recessive or dominant, and also sex-linked also it can be recessive or dominant. So here, if we talk about the Mendelian disorders, these Mendelian disorders are of two types. One is autosomal, and another one are the sex-linked, or we can say X-linked. If they are autosomal, then in autosomal they can be dominant or they can be recessive. Means the genes is present on the autosome, but in dominant condition, if it is showing a disorder, then it is autosomal dominant disorder, and if it is showing in a recessive condition, then it is a autosomal recessive condition. Same way in case of, we can say, X-linked, sex chromosome linked or sex linked inheritance or X linked inheritance, you will see the X linked recessive disorder and X linked dominant disorder. Mainly here we are going to discuss autosomal dominant, autosomal recessive, or mainly autosomal recessive, and X linked recessive disorder we are going to discuss. Okay, so first, if we discuss about this disorders, we are going to talk about gene mutation in autosome. Okay, the disorders due to gene mutation in autosomes Then in this, the mutation can be on a recessive or can be dominant. So we are going to first talk about autosomal recessive disorders. Then we will discuss autosomal dominant disorders. Autosomal recessive disorders are important. Now, these autosomal recessive disorders are caused by a recessive autosomal gene. These are caused by recessive autosomal gene when present in homologous when present in homologous condition. So here, if we talk about autosomal recessive disorders, they are as follows. And if we can talk about the examples of autosomal recessive disorder, then first is alkaptonuria. Second is albinism. Third one is Tay-Sachs. This is fourth. And Kocher. This is fifth. Sickle cell anemia. Sixth, thalassemia. Seventh, cystic fibrosis. And eight, phenylketonuria. So these eight type of disorders are mainly found in case of the autosomal recessive disorder conditions. In this, some are important, like alkaptonuria, albinism are important. We will discuss that. Sickle cell anemia is important. Cystic fibrosis is important. Thalassemia is important. Phenylketonuria is important. So these important disorders we are going to discuss in detail. So first, we are going to discuss alkaptonuria. Now, when we talk about this alkaptonuria is the first inborn metabolic disease. It is a inborn error of metabolism. Means it is inborn, means it is present by birth, and it is inherited in autosomal recessive condition. Now, this metabolic disorder is produced due to this metabolic disorder is produced due to deficiency of an oxidase enzyme required for breakdown of tyrosine. Now, why this oxidase enzyme is absent or deficient? Because there is a mutation in the gene, and when this mutation convert this particular gene in a dominant and a recessive condition, and when both the genes are in a recessive condition and in a homozygous condition, and they are not able to produce this oxidase enzyme, then this breakdown of tyrosine will not occur. So tyrosine is breakdown is not going to occur due to the deficiency of oxidase enzyme. So its toxic product homoxanthesic acid, also called alkapton, accumulates. And when this toxic product accumulate, the disease is known as, because of this disease known as alkaptonuria. Now, this is due to absence of uh, normal form of gene on chromosome 3 that control the synthesis of enzyme. Now, this homoxanthesic acid accumulate, and when it accumulated in the tissue, it is also excreted in urine. So it accumulate in tissue and also excreted in urine. Now, when it is excreted urine, what is going to happen? So, if urine of this patient urine of this patient, if allowed to stand for few hours in air, it this urine turn black in color due to oxidation of homoxanthesic acid. So when there is a capital A condition, there is a gene are in a normal condition. But when small a is condition, then it is going to be defective problem or recessive condition, and in this the particular this oxidase enzyme get deficient and this alkaptonuria occurs. Okay, so this is the first disorder which is observed. After this second disorder, so see, one of the important thing is whatever the points I am considering, that are the important points. Okay, so what are the things can be asked according to Neet, I will cover that only. The another disorder is albinism, and albinism is considered as also autosomal recessive disorder. Now, it is also inborn error of metabolism. Now, in albinism, this is caused by absence of enzyme, by absence of enzyme tyrosinase. So, if there is a small a condition, it is not going to produce tyrosinase. And when tyrosinase enzyme is absent, which is essential for melanin, essential for the synthesis of melanin pigment actually, from the dihydroxyphenylalanine. So if this enzyme is not there, this enzyme not going to function, and this dihydroxyphylline is not going to convert it into the melanin because the enzyme is deficient and enzyme is required. So recessive allele do not produce the enzyme, but normal allele can produce. So now, due to the deficiency of melanin, albinos are produced. So albinos means individual with albinism, like this dark pigment melanin in skin, hair, and iris. Now this albinos, although have a poor vision, have poor vision, but they lead normal life. Okay, so that is the albinism. After albinism, third disorder, which is autosomal recessive, that is in genetic, that is tie such disease, that is TSD. If we talk about this tie such disease or TSD, it is also autosomal recessive disorder, and this is the disorder occur due to defect in lipid metabolism. And in this, the homozygous recessive children show a degeneration of the central nervous system due to accumulation of the some fatty substance that is known as sphingolipid in nerve cell. And the children with this Tyson disease are born normal, but they develop severe brain and spinal cord damage later in few months due to the error on metabolism. So they are mentally retarded, progressively paralyzed, child within 3 to 4 years, and there is no treatment for this, and hence no survivors of this Tyson disease are known. So more details are not going to be asked for this disease, but you should remember that this is the defect of lipid metabolism. And actually, this the earlier two we have studied, like alkaptonuria and albinism, they are the defects of amino acid metabolism. Okay. After this, the fourth disorder, that is Gaucher's disease. This Gaucher disease is also a defect in lipid metabolism. And there is no need to discuss this much more because no one is going to ask a question from this. But in Gaucher disease, it is also a lipid metabolism defect, and in this disorder the breakdown of fatty substance cerebroside does not occur that lead to the accumulation of this material, fatty material or lipid material in body tissues and blood, and this inhibit the activity of enzyme glucocerebrosidase in lysosome. So accumulation of this cerebroside occur, and the enlargement of spleen and liver will be shown. So this can occur in the Gaucher disease, but more detail are not going to, no need to remember. But after this, one of the important diseases we are going to discuss, that is the fifth one, and that is sickle cell anemia. One of the important disorder and most commonly asked question. Okay, so here it is a sickle cell anemia. Okay, we have like sickle cell anemia. Okay, here it is also autosomal hereditary recessive disorder. In this, erythrocytes or RBC become sickle shaped, under oxygen deficiency. under oxygen deficiency. As you know that during a strenuous exercise and at high altitude, you know that there is oxygen deficiency, and during with this condition, this RBC is going to be sickle shape. And this oxygen deficiency occur because this disease or disorder is caused by formation of abnormal hemoglobin, formation of abnormal hemoglobin known as hemoglobin S. Now this abnormal hemoglobin, hemoglobin S, if we talk, these are modified from the hemoglobin A. Now, if we compare this hemoglobin S from the hemoglobin A normal, only one amino acid change is observed. And here, glutamic acid is replaced by valine at the sixth position of the beta acid. Now, how this occur? If we want to discuss that, let's discuss. So, in this, if we talk about normal hemoglobin A gene, then in DNA, you will see that there is GA, G and CTC. Now when it get transcribed, GAG occur in 5' to 3' condition. So this CTC should be in 3' to 5' and this is in 5' to 3'. So CTC complementary GAG is synthesized in the mRNA. And when this mRNA transcribe, it is going to add glutamic acid at the sixth position. Second amino acid, third amino acid, fourth amino acid, fifth amino acid, and the sixth amino acid is going to be glutamic acid. But when the sickle cell hemoglobin is gene is there, then in DNA you will see that there is a GTG and CAC. Now when CAC get transcribed, that is GUG in 5' to 3' a gene, and this codon is present on mRNA. So at the sixth position, it is going to code for the valine. Okay, so at the sixth position, hemoglobin S in amino as polypeptide, the glutamic acid is changed to the valine. The mutation in DNA, you will see just that in this chain, the T is replaced by A. So when T is substituted by A, this is known as transversion, and due to this transversion mutation, the hemoglobin A become hemoglobin S. Okay, so here this hemoglobin S is produced due to change in codon codon GAG to GUG on mRNA due to substitution of T by A on DNA. So now the GAG converted into GUG on the mRNA. So GAG code for the valine. So here glutamic acid is replaced by the valine, and these show the particular this distort the structure. So here this transversion mutation of the gene controlling beta chain of hemoglobin, the mutated gene is called hemoglobin S. So hemoglobin S causes one change in amino acid sequence of beta chain. It replaces glutamic acid at sixth position of the beta chain by amino acid valine. The mutant hemoglobin molecule undergoes polymerization under low oxygen. So mutant hemoglobin undergo polymerization under low oxygen tension, causing change in shape of RBC from biconcave disc to elongated skull-like structure. Okay, so if the gene which are controlling this or a genotype in a population are in HbA condition, then it is a normal homozygous condition. Whereas if the HbA, HbS genotype are there, then individual are normal but carrier or sickle cell gene. But if there is a HbS, both the alleles are in a diseased condition or modified condition or hemoglobin S condition, then this is the diseased, and this individual going to die before attaining maturity. Okay, so this is the thing observed in case of the sickle cell anemia. So if we make a cross of carrier woman, that is like HbA, HbS gene will be there, and if it is a carrier man, it is going to HbA, HbS condition, and if we make a cross, and these are if we take a parents, then gametes are going to be like this, that HbA, HbS. Here all also gamete are going to HbA, HbS. Now if we make a cross and we find an offspring, then if here we take HbA, HbS, HbA, HbS, then here it going to be HbA, HbA. Here it is going to HbA, HbS, HbA, HbS. HbS, HbS. So this HbA is then going to be normal hemoglobin. This is the carrier individual. This is also carrier individual, but this is going to die before childhood. This is sickle cell anemic. Okay. So here the ratio of 1 is to 2 is obtained for normal is to carrier. Okay, so this is the condition is seen in case of the sickle cell anemia. So this is the condition of a sickle cell anemia and what is sickle cell anemia that we have discussed. Now we are going to discuss another one, that is thalassemia. And if we talk about thalassemia, it is also known as Cooley's syndrome because it is discovered by Cooley. Okay. Now thalassemia is autosomal recessive blood disease and this defect occur due to mutation or deletion of a genes controlling the formation of globin chains. Okay, so here globin chains are going to produce less, and which globin chains commonly alpha and beta of hemoglobin are going to be produced less. So this imbalance synthesis of globin chain of hemoglobin causes anemia. Okay, so anemia is the characteristic feature of this disease, and depending upon the globin chain affected, depending upon the globin chain affected, thalassemia is of three types, and that is alpha, beta, and delta. So now let's discuss alpha, beta, and delta thalassemia. Now, first, if we talk about the alpha thalassemia, in alpha thalassemia, what happens? This is caused due to the defective formation of alpha globin, or we can say the deficiency of alpha chain, but defective alleles of the alpha globin. So in alpha thalassemia, here it involves the genes HbA1 and HbA2. They are inherited in a Mendelian recessive fashion, and they are two gene. So they have four alleles. Okay, And here in this deletion, these are what happens here. Deletion in 16p means short arm of 16 chromosome. So if there is a deletion in the short arm of 16th chromosome, the result in decreased alpha globin production. And due to the fewer alpha globin chain produced, this result in excess of beta chain production in adults and gamma chains production in newborn. Now this excess of beta chain form unstable tetramers called hemoglobin H or HbH of 4 beta chains. Okay, and this will lead to the abnormal oxygen dissociation curve. Okay, so in alpha thalassemia, alpha chain are going to be produced less, but beta chain production is increased in case of adults and gamma chain production increase in case of the newborn. But when we talk about the beta thalassemia, in this occur due to mutation in HBB gene on chromosome 11. So the gene in which mutation is going to occur for the beta thalassemia is present on chromosome 11, and it would also get inherited in the autosomal recessive fashion. Now in this, the beta chain production become less. So beta chain production decreases, and alpha chain production increases. And here this increased alpha globin chain do not form tetramers, but they bind to RBC membrane and cause membrane damage. And at high concentration, this alpha globin form toxic aggregates. Okay. Now the mutation in this HBB gene is can be minor. That is the we can call this particular individual which having such type of minor mutation are beta thalassemia minor Okay, but if there is a what particularly we can say the affect any formation of beta chain, then it is there is no formation of beta chain, then it is a thalassemia major. So if it is a B0 or beta thalassemia major, so it can occur if they prevent any formation of beta chains. Means beta chain is not going to produce at all, and this beta thalassemia major is a severe form of beta thalassemia. But B plus or beta thalassemia intermedia, if we talk, this in intermedia, they allow some formation, some beta chain formation. Okay, in this both case there is a excess of alpha chain, and what alpha chain causes that we have discussed. Now third type is delta thalassemia. And in delta thalassemia, just like beta thalassemia, the mutation can occur which affect the ability of this gene to produce a delta gene. So here in delta thalassemia, delta chain production decreases. Now in human, if we talk, 3% of adult hemoglobin is made up of alpha and delta genes. So if there is a decrease in production of delta chain, the alpha gene production may get increased. The mutation will lead to the what decrease in the alpha chain, delta chain production. So this thalassemia, the production of globin chain is decreasing, but in sickle cell anemia, the defective globin chain is going to produce. So here it is a, or thalassemia is a quantitative disorder. But in sickle cell anemia, the defective globin chain are produced, so that is a qualitative disorder. Okay. Now, after this, we are going to talk about the cystic fibrosis. In cystic fibrosis, if we talk, it is also autosomal recessive disorder of infant, children, and young adults. So here, the gene which is present on chromosome number 7, gene on chromosome 7 become recessive. When this gene present on the 7 become recessive, that is due to the deletion of 3 bases, then this disease occur. Now this disease gets its name due to formation of fibrous cyst in pancreas. So it produce defective glycoprotein. And when there is a defective glycoprotein production, it causes thick mucus formation in skin, lung, pancreas, liver, and other secretory organ. So the sweat of patient contain very high level of Na plus Na plus and Cl minus. So there is a failure of chloride ion transport mechanism in cell surface membrane of epithelium cell. Now this accumulation of thick mucus in lungs and airways, they block this. Then there will be a recurrent pulmonary infection and irreversible lung damage. Further, the mucus deposition in the pancreas blocks secretion of pancreatic juices. The maldigestion of food will happen with high fat content in the stool. Liver may undergo cirrhosis and the impaired production of bile. Now, how we identify that this disorder is there? So, this disorder identified due to the what sweat of sufferer is saltier. So, if we kiss a child, then it tastes salty. Okay, then we can identify that this is the cystic fibrosis. After cystic fibrosis, the next disorder which we are going to discuss, that is phenylketonuria. If we talk about this phenylketonuria, it is a inborn error of metabolism. It is also inherited in autosomal recessive fashion. Now, affected individual, if we talk, lack and liver enzyme, that is phenylalanine hydroxylase. When there is a deficiency of phenylalanine hydroxylase, which convert amino acid phenylalanine to tyrosine. If the enzyme is not there, then there is no conversion of phenylalanine to the tyrosine. And as a result, phenylalanine accumulate and converted into phenylpyruvic acid and other derivatives. Now, accumulation of this in brain, its accumulation in brain result in mental retardation. Okay, and these are excreted. These are excreted through urine because of poor absorption by kidney. Okay, so that is the phenylketonuria, and that is all about the what we can say autosomal recessive disorder. Now we are going to discuss some of the autosomal dominant recessive disorder. In this, we no need to discuss in detail. Just we have to list it. Okay, and some point we need to note down. So here we are going to talk about autosomal dominant disorder. So if we talk about the autosomal dominant disorder, they are caused by dominant autosomal genes, and some of these dominant autosomal inherited disorders in human being are. So first disorder is achondroplasia. Achondroplasia is a dwarfism of long bones. Dwarfism in which long bones do not grow. Second is polydactyly. And in polydactyly, there is a presence of extra finger or toes. Then third autosomal dominant disorder is brachydactyly. And this brachydactyly means abnormal short finger and toes. Fourth is Huntington disease. Or Huntington chorea. This is important as you should remember that is autosomal dominant because it is a disorder in which muscle and mental retardation occur. And due to the gradual loss of motor control resulting in uncontrollable shaking and dance like movement. Chorea means dance like movement. So if a person is going walking, just it is walking, you will see that it is walking like a dancing person. In this brain shrinks, okay, and there it will be followed by a slurring speech, loss of memory, sometimes hallucination occurs. And this disorder does not occur till the age of 25 to 55. Okay, so this defective gene is present on the chromosome 4, and here the frequency is 1 in 10,000 to 1 in 20,000 individuals. Then fifth is PTC, that is phenyl thiocarbamide testing. So the dominant are only going to be testers. Okay. Then myotonic dystrophy. And if we talk about this myotonic dystrophy, it is characterized by increased muscular irritability and contractibility with a decrease in the relaxation. So contraction is increased, but decrease relaxation is slower down. So when contraction increases but relaxation does not happen properly, then this leads to the atrophy of muscle, especially of face and neck. Okay. such thing occur in case of myocardial dystrophy. So that is also autosomal dominant disorder, and it is a inheritable disorder. Okay. Now after this, we are going to talk about the gene mutation in sex chromosome or sex chromosome sex-linked disorders. We are going to okay. So we are going to sex-linked recessive disorders. Only recessive disorders we are going to discuss here. So now we are going to discuss X-linked recessive disorder. In this X-linked recessive disorders, we will see that when the genes which are present on X chromosome and they are in a recessive condition, they are going to cause a disease, and they are getting inherited from particular parents to offspring. And in this first disorder, which we are going to discuss, that is hemophilia. Now, when we discuss hemophilia, it is a sex-linked disease which is known as bleeder disease. It is also known as bleeders disease. And here, as the patient will continue to bleed even after minor cut. In hemophilia, what happens? If a minor cut is going to happen, then after that also, the patient will going to continue to bleed even from this minor cut, since he or she does not possess the natural phenomena of blood clotting. Okay, so natural phenomena of blood clotting does not occur immediately, so the bleeding continues. So this is hemophilia occur due to absence of anti-hemophilic anti-hemophilic globulin factor or factor 8. And if it occurred due to the absence of anti-hemoglobin factor 8, then it is known as hemophilia A. And if it is due to absence of plasma thromboplastin factor 9, then it is known as hemophilia B or Christmas disease. Okay, so as a result of continuous bleeding, as a result of continuous bleeding, patient may die of blood loss. Actually, in hemophilia, blood does not clot within 3 to 8 minutes. It takes approximately 1 hour or more. Usually, it should clot within 3 to 8 minutes, but it takes hours. Because of this, there is a chance of death. In this hemophilia, if we talk, there is no permanent cure. or of this disease. However, transfusion of normal blood check or prevent bleeding, because this transfusion of blood will going to provide the vital factors or clotting factors which are required for the blood clotting. Okay, so anti-hemoglobin factor will also be available for the use in hemophilia. Okay, so this hemophilia is genetically due to the presence of recessive sex-linked gene H. Okay, so it is due to recessive gene H on X chromosome. So if it is X, H, and X condition, then it is a carrier female. But if it is a XH, XH condition, then it is affected, or we can say hemophilic female. But if it is XHY, then it is hemophilic male. So such representation are used for the indication of this disease. And this hemophilia disease is also known as royal disease. Also known as royal disease, because it has been quite common in the royal families of Europe, and the disease spread to them through the children of Queen Victoria. So the ancestors of the Queen did not possess the disease. It appears that the gene for hemophilia developed either in the germ cells of her father or herself through the mutation. Father of Queen Victoria or the mutation in the. So being a sex link, the gene for hemophilia shows a crisscross inheritance. This disorder shows a crisscross inheritance. Yes, it can be transmitted from mother to the son to the daughter. And the frequency of this disease is frequency is 1 in 7,000 human males and 1 in 1 crore, or a case we can say 100 lakh in females. Okay, so now if we make a cross and we take an example of this inheritance, then if we take a carrier woman, if we make a cross with the normal man, in carrier woman you will see that XX condition, in normal man you will see XY condition. So here the gametes are going to be XH and X type, but here gametes are going to be X and Y type. So if you make a cross here between these two, then if you make a cross of XH and X, with this X and Y, then XHX will be there, XX will be there, XHY will be there, and XY will be there. So it is a carrier female. Okay, this is a normal female, this is normal male, this is a hemophilic male. So here one normal girl, one carrier girl, one hemophilic boy, and one normal boy and one hemophilic boy is obtained. So in 1:1:1:1 condition is observed. If similarly, if make a cross, another cross like this of a normal female with hemophilic male. If it is normal female, then it is represented by XX, and if it is a hemophilic male, it is represented by XHY. So here the gametes have only X chromosome, here the gametes have XH and Y condition. So if this X crossed with this mate with this one, then X going to occur in the offspring. And if it fertilizes with this one, then XY going to occur in this particular offspring. XHX condition if we talk, then it is going to be the carrier girl and it is the normal boy. And these both are going to in a 1:1 ratio. Okay. So this is the what a particular hemophilia or bladder disease. After this, we are going to talk about another disease that is a color blindness. If we talk about color blindness, it is also known as red-green color blindness. Okay, and in red-green color blindness, here the sex-linked recessive gene are occurring, which here you will see the person suffering from color blindness whose eye fails to distinguish red and green color. And due to this, you will see that vision is, however, not affected, and the colorblind person can lead a normal life. They can reading, writing, driving, all they can do. But in traffic signals, they distinguish the traffic light by their position. But gene for normal vision is dominant. But for a recessive allele on an X chromosome, it is going to be affected. So female color blindness will occur if this is XC, then it is a color blind female. If it is XCX, then it is a carrier female. If it is XCY, then it is what we can say colorblind male. Okay. By using this condition, you can continue to make a cross for the this color blindness. Now, after color blindness, we are going to continue the night blindness. In night blindness, if we talk, it is caused by a recessive gene carried on X chromosome. This genetic disorder is also more common in males. It is of two types actually. Okay, one is acquired. Okay, that is due to vitamin A deficiency. Another one is the congenital, that is due to visual purple pigment deficiency. Now, due to this deficiency, due to visual deficiency, and that interfere with function of retinal rods. It is inherited like a red-green color blindness, and here female become night blind only if there is two genes of the defect. So inheritance is going to be like the what? A color blindness. Now fourth disorder, which is X-linked recessive disorder, that is Dutchne muscular dystrophy. In Dutchne muscular dystrophy, if you talk, that is DMD. In this disorder, the mutated gene, that is dystrophin gene, it is present in middle of short arm of X chromosome. And here, this mutated dystrophin gene is the largest gene, that is 2400 kilobase pair in man. Now, if the mutation occur, then this mutated gene is unable to produce dystrophin protein in skeletal muscle. And this dystrophin protein actually it relay or pass nerve signal to calcium storage in muscle cell. Okay. Now, the function of this is to pass the nerve signal to the calcium storage in the muscle cell. Now, one more important thing is that this protein this dystrophin protein is associated with plasma membrane of muscle, that is sarcolemma. And this protein here play a role in transmitting signal and stabilizing the membrane. Now, due to this deficiency, due to its deficiency, calcium is not released from muscle cell. And when calcium is not released from the muscle cell, as a result, muscle contraction does not take place. And when muscle contraction does not take place, so this lead to the abnormal rise of calcium level in muscle. And this abnormal rise in calcium level in muscle, it releases enzyme that destroy actin and myosin, and due to the destruction of the actin and myosin, it result in fatal muscular weakness. Okay, so due to this fatal muscular weakness, the patient is unable to walk after age of 12. That lead to later on mental impairment and death by age of 20 due to respiratory failure or cardiac failure. So this is the this is about the H&M muscular dystrophy, and that is all about the sex-linked recessive disorders. So now here we have completed with the Mendelian disorders, and whatever the important disorders are there, we have discussed that. Okay. So I hope you understood. Thank you.